about branching morphogenesis.
Thank you.
So, thank you. Yes. In the spirit of the last talk of the day, it is the last talk, isn't it? In the spirit of the last talk of the day, my talk at least will be simple, I think. So that's good. The bad thing is that it's kind of the wrong order with. So Dagmar gave a very beautiful talk with all of the complexity, which you will wonder why it's missing from my talk, and it probably should be there. So anyway, the problem I want to talk about is branching morphogenesis. The focus of the talk will probably be is largely centered on mammary gland epithelium, but some of the principles that emerge from that seem to be common to other tissue types. So we can discuss that, which is why I called it a unifying theory. And I suppose in the spirit of the workshop, I suppose it should probably also give my kind of philosophical take on the field, but of course it will rather strongly overlap with what other people have said. So like so many people here, we're interested in trying to understand how regulatory programs encoded at the molecular scale integrate together into developing the gross organization of tissue at the organ scale here in the case of the pancreas. We know that cell fate decisions are mediated by interactions of genes and gene products, and we know that the large-scale structure of tissue is organized by collective cell movements and cooperative fate decisions. So, of course, the obvious question to ask, if you're a physicist, is what's the right language to articulate mechanistic understanding? But in the developmental biology community, or cell biology community, this isn't really a question, because the answer is clear. It's, of course, molecules. And I'm interested in that too. So fundamentally, of course, we would like to understand what's happening at the most microscopic, basic level to affect fate decisions. But if you're trying to understand something like the large-scale patterning of tissue, you have to ask yourself, are these always the right starting variables? So I suppose the kind of philosophical lesson I want to try to convey in this talk is that for at least the example we're going to consider, this might not be the best reference frame to ask the kinds of questions that we want to ask. So in my group, or in my lab, we're interested in trying to understand how emergent collective behavior can provide predictive insights into cell-fate decision-making in both development and adult tissues. And of course, our target, like everybody else, is to understand how these insights can produce for us targeted questions into molecular regulatory programs. So that is our target, but we want to start, I think, with what in physics we call phenomenology. So the problem that I want to consider, and we'll see how far we get through it today, is the problem of branching morphogenesis. So, of course, to sustain life, organisms have to exchange nutrients and waste products with the environment. In unicellular organisms, this can be achieved just at the level of a single cell, but in multicellular organisms, strategies have to be found to maximize the area of exchange surfaces. So, for example, here in the small intestine, the epithelium divides up into villi, which invade into the lumen to increase the surface area. In three-dimensional organs, like kidney, lung, mammary gland, as you've seen today, these exchange surfaces end up packed around highly ramified, complex branched epithelia. And so the question that I want to try to address is how does all of this information encoded at the microscopic scale translate into large-scale organization and patterning at the organ scale? And the workhorse for this question is the mouse mammary gland. So the mammary gland in mouse is specified along the ventral epidermis around E11 as placode-like structures that embryonically invade into the fat pad, into this adipocyte-rich fat pad region. And around birth, you have a rudimentary tree-like structure. And then during puberty, this rudimentary tree-like structure evolves into a highly complex, ramified ductal epithelium. And then in adult, through estrous cycle, there are bouts of growth of alveoli which grow and regress. So this growth is generated, is driven by populations of stem cells that reside in the tips of these terminal end buds, And the mammary stem cells, they drive a process of sequential bifurcation and ductal elongation, producing this highly ramified network. So what we've also heard before today is that the ductal network that they leave behind is a simple stratified epithelium involving myoepithelial basal cells and luminal cells. So if we look, here are several fat pads during pubertal development. This is 3.5 weeks. This is the origin of the branching process. Here you can see stemmed with basal ductal marker K14. You can see the emergence of these ducts. At 5 weeks, they've grown this far, and by end of puberty, they've grown to expand across the entire fat pad. So another simple observation within the system is if you stain with the proliferation marker EDU, then here you can see tips which are EDU positive and highly proliferative, and all EDU is incorporated only into the tips. There are no side branching in this system. So as the cells leave the tips, they exit cell cycle. They're still capable of proliferation, but they're non-proliferative. And we also see that there are tips which are EDU negative. So we know that the stem cells are restricted to tip region. So the question I want to ask is, how do stem cells integrate their fate choice with all these collective cell movements to define this large-scale patterning of the network? What is the identity of the mammary stem cells? What's the multiplicity? Are there all stem cells? Is it just one? Is it just a few? And what's the sublineage potential of those mammary stem cells? Are they bipotent, able to generate basal and luminal cells, or are they lineage-restricted? Are they stem cells at all? This is a developmental context. Maybe this is not a right language to describe these cells. And finally, are the principles of patterning that emerge from this tissue, are they conserved in other tissue types? So where to begin? So what's the right level of abstraction? And as you can imagine, this is a very intensely studied question in the field, and most of the attention is placed on trying to resolve the repertoire of signaling pathways and expression signatures of the cells and the tips of the end buds. But actually, we're going to focus on what's happening at the large scale in this system and see how far that gets us. So the experimental work, this part of the work was done in collaboration with Jaakko van Rienen's lab. Some of you may know Jaakko's lab. He's a specialist in intravital light imaging, and when we began this collaboration, he was at the Hubrecht Institute. He's now moved to Amsterdam. And all of the work, all of the heavy lifting was done by a fabulous student of his, Kalinda Shida. So here in Holmount is a mammary fat pad stained with keratin 14, which is marking the ducts. And what Kalinda did was take these fat pads and segment them. And here is a typical segmented ductal network. And then what she did was that she looked at the topology of that network, one after the other. I can't remember whether that network is that one, but you can imagine she did this for large numbers of different ductal trees. And what you can see at first sight is slightly surprising. You can see that there are some small subtrees, and there's some very large subtrees, and there is enormous heterogeneity. And the second thing that was clear is that if you look at the frequency of end buds that are still proliferative, they gradually diminish during development. So you have active end buds, but the frequency of those end buds is gradually declining through puberty.
Everywhere in space, or in some locations in space?
You'll see. I mean, for now, think of it as statistics, but of course this will be regionally localized in a way which would need to be disclosed what's going on. Let's not spoil the suspense. So where does this complexity come from? Is it coming from early specification of stem cells which have a defined but variable proliferative potential? Doesn't sound very likely, of course. Or are there extrinsic factors that are influencing the fate decisions of what are otherwise equipotent populations of stem cells? So to try to address this question, the first thing we can ask is whether there's evidence for equipotency. So we just looked at a set of morphometric measurements we looked at the width of the ducts and the length of the ducts as a function of the branch level. I hope the notation is clear. So each time you branch, you move up one more level. And you can see that after the specification of the rudimentary tree, both the width and the length of the ducts, on average, are relatively uniform. And if we look at the proliferative activity of active ducts, so if they're active, how much EDU do they incorporate? And they're highly proliferative and hardly changing. So either all of the cells are rapidly cycling in the end bud, or they've stopped cycling altogether. There's no gradual dilution of the EDU expression. So everything looks highly equipotent. It looks like the buds early in development that are active look just as potent as they are later in development. So what is the source of heterogeneity? So what we realized is that you can take a network like this, and you can obviously segment it into a sequence of decisions where an active n-bud will either bifurcate with probability 1 minus q, or it will terminate through cell cycle exit. Obviously, you can take any network and present it in this way. But we speculated that this might be a random decision, with q just changing according to the branch generation number. So we just empirically measured that for lots of n-buds. And here it is. So you start off tilted towards low q, which means a high rate of branching. But as you move through development, this sort of heads towards a plateau around a half. So it looks like every time you have an active n-bud, it flips a coin. If it's heads, you branch, and if it's tails, you bifurcate. Well, this is purely empirical, and we can always do this, but we can ask, can we predict something with this? So if we take this kind of dynamic, can we predict something about the full distribution of the network? So we can predict what is the fraction of active n-buds over time. The points are experiment here, and the line is what comes out of this very empirical model. But we can also predict the size distribution of the different subtrees, or the chance that a subtree persists over time. And it isn't too bad. This is, in some sense, it's a no-parameter model because you just measure this and you ask, can you predict that? It's not perfect. It's actually not too bad. So it looks like the entire topology of this network can be predicted by a simple statistical rule, which is active at the ductal scale.
It would never stop growing, but if Q is 0.5.
Keep the thought. You're absolutely right. Yes, keep the thought. I'll come back to it. So what regulates this convergence towards near balance of stochastic ductal bifurcation and termination? And in fact, the reason is obvious. Yes?
Just a pure curiosity, do you ever see more than bifurcation? So 3 or 4?
It's getting very annoying, and I should write the question. Please could you also keep that question? Right? Because you'll see that the answer is quite subtle. Okay. But if I forget to answer the question after my time, before my time is up, you'll remind me. It's you'll see why I said that. Okay, so what we noticed, and it's very clear here, so branching morphogenesis in the mammary gland takes place in a largely two-dimensional setting. And it's very clear that there are very few crossovers. In fact, we can score the frequency of crossovers, and it's tiny. And the density of these branches is roughly constant. So what we suggested was that maybe the way this tissue develops is through the process of the branching annihilating random walk. So the idea here is that the terminal n-buds are continuously driving tip elongation. They're continuously and stochastically branching. We have no idea why they branch. And actually, Dagmar might have an idea about why they branch, but I'm not going to, it's a great question. We have no idea why they branch. We say they stochastically branch, they elongate, but when an active tip moves into proximity of a maturing duct, the cells exit cell cycle. So this is a branching annihilating random walk. It's actually been considered by many people, including Meinhardt early on, and we'll come back to it. So if we run a kind of toy simulation, then here is such a branching annihilating random walk, and when this random walk hits the edge of the fat pad, then it terminates. So there are signals at the edge of the fat pad to terminate it as well, which is the question. So it kind of looks okay. I mean, it's a toy. It kind of looks okay. But what kind of predictions can we make with that? Can we actually make quantitative predictions of the branching distribution? So here's a typical simulation. Here's a typical experiment. And the first thing that we noted was that for the simplest possible model, we suppose that the timing between consecutive divisions was random, just for simplicity. Actually, if you measure experimentally the size distribution of the ducts, the lengths of ducts, it's actually astonishingly Poisson distribution-like. It's astonishingly exponential. So that goes to the question that you asked about whether you only get bifurcations. The maximum probability is for a bifurcation of length 0. So we see some events that you might think is a trifurcation, but it's sitting nicely on this distribution. So we can take these simulations and we can segment them, and we can do the same with the experiment, and we can ask, what do we predict? And this is what we predict. So in this model, there is one parameter, which is the ratio of the branching rate to the elongation rate. But we don't fit that, we just measure it because it's connected to the tip density. So this is a no-parameter model, which is predicting extremely well the change in the branching probability over time. It's predicting extremely well the subtree size distribution and the subtree systems. Yes?
Is this in 2 dimensions?
Yes.
Okay. And that makes sense for the experiments?
Yes.
Okay.
Yeah. It takes place, it's a very two-dimensional setting. So are there any analytical insights that we can use to challenge the viability of this model? And actually, it's quite an interesting problem. So the problem of branching and annihilating random walks has been considered by many people. Notably, John Cardy has been all over this problem. And actually, the place where it's been most sort of thoroughly understood is in the context of tip-tip annihilation. But that's not the context that we've got here. In fact, it's much closer to an earlier work that John Cardy did with Peter Glasberger on epidemic models and percolation. And it's a much more complex kind of system. And I don't want to get way down in this because it's honestly, this part is not so interesting, but it is worth mentioning. So what's the connection with epidemic models? So in fact, this branching annihilating random walk is very much like an epidemic model in the sense that the active tips are like infectious agents that diffuse and can multiply, which is the process of ductal branching. And what they do is they leave behind a trail of infected sites as they move over the system. But those infected sites can recover, and if the infectious agents meet again those recovered sites, they terminate. So this was the model that Cardy and Grossberger considered, and what they noted, well, they wrote down an action for this non-equilibrium system. It's got a critical dimension of 6. And this model describes a phase transition between a phase in which you inevitably terminate by colliding on the network and a phase in which you have a chance that you can extend out to infinity. In the physical system, we're very far from this phase transition, and so we can use beam field approach to try to understand what's going on, and that's what I want to focus on. So at the level of mean field theory, the branching dynamics can be expressed through a concentration of active tips and a concentration of inactive ducts. And at the mean field level, the active tips are diffusing, they're increasing in number through branching, and they're disappearing through annihilation with inactive ducts. And the inactive ducts are immobile, they don't diffuse. They're generated through the elongation process from active ducts, and they're also generated in the annihilation process. And if you rescale out the dimension for parameters there, you end up with the following system, which indeed depends on just that one parameter, the ratio of the branching rate to the elongation rate, then that system is a Fisher-KPP reaction diffusion system. But what it describes is a front which propagates at constant speed across the network, a front of self-organized active tips. So we can see that if we solve the Fisher-KPP equation, then what we can see is that the active tips organize into a soliton-like pulse that moves at constant speed, which is connected just to this parameter, that moves at constant speed, leaving behind a uniform density of inactive ducts.
What is known experimentally about the rate of annihilation compared to the rate of bifurcation or branching? In other words, the dynamics of your A compared to Y, are they comparable?
Yeah, okay, so this is a good question. We can actually, so we can infer this parameter directly. I think the right way to articulate the answer to this question. So we can infer this parameter R, but it's a dimensionless parameter. So in a way, what you're asking is, is it of order 1? And the answer is it is of order 1. So now we can understand that very simple phenomenology that we had in the beginning. This is a system where there's density-dependent feedback. So if you considered an active tip, which by chance found itself in the center of this network, it would annihilate almost immediately. On the boundary, every time a tip bifurcates, on average, one new tip pioneers virgin territory and the other collides with the trailing network, which is why the system converges towards this critical light state where you have this 50-50 balance between bifurcation and termination.
That must be very dimension dependent.
No.
In 3 dimensions, you
Same. Yeah.
same.
Yeah. At the level of mean field.
Okay.
So we can test this, actually, which was really pushing this model to the very limit. So here is, again, experimental data. The red is EDU. And again, we took multiple glands and segmented those. And here you can see the data for the uniform density of the trailing network. And here is the density of active tips, which is this putative soliton-like pulse. And actually, one of the nice things that emerges from this is if you look at the exponent, you've got an exponential decay on both sides of this. If you look at the ratio of the exponent, it's just a number. And actually, it works pretty well on the experiment. This is not our data, but you can look at the speed at the front as it moves across the fat pad, and it's fairly linear over time, which is, again, a hallmark of this Fisher-KPP-like dynamics. And another thing that we can look at, this is a non-equilibrium system. So if we look at density fluctuations, we'd expect to see a non-trivial exponent. So in other words, if we look at the variance in the density of ducts as a function of the average density of ducts, then what we get both experimentally and from the numerical simulations is an anomalous exponent here that's close to 2/3. So what we have is giant density fluctuations in this system, and they emerge because it's a bit like DLA in fractals, because you can create these large voids that once they close, there's no way they can be filled. And the other puzzle in the Mammary field was why you have this kind of directed growth through the fat pad. And people for a long time had looked for morphogens that might guide the branching process as it fills the fat pad. But if we measure the angle probabilities of the ducts across the fat pad, then we can actually see that the model predicts very well that directional bias, which just comes from the symmetry breaking due to the nature of the growth process. In other words, we have directed growth without any morphogen gradient. Right, so at the large-scale level, what's driving, just one second, so what's driving the large-scale organization, we would argue, is just density-dependent feedback. You have this process of ductal elongation, stochastic bifurcation, but when active tips meet maturing ducts, they just stop. Yes?
So that angle you were mentioning was the angle of each bifurcation?
Okay, so again, there's a very good question, which I anticipate will be asked later on. Does the particular angle of bifurcation matter? And this is very good. There are multiple ways that one can start refining the model. In the particular concepts of the Mammary gland, where the parameters sit, we see that the statistics don't depend very much on what that bifurcation angle is. But you could find a different part of the phase space where it would matter. So the angle there was just looking at the sort of gross angles of the ducts from the experiment, but not necessarily the specific branching angle.
And in this elongation, is it just a straight elongation, or it's also random?
Well, no,
walking?
indeed this is the same question. So as you tune this dimensionless parameter R, you would transit from having very straight segments to highly meandering segments. From the experimental data, we get the R value that we get, and they're relatively straight. There's not much time for these to do a lot of meandering.
The question is the fat pad really just pure fat, or is there other stuff in there that the tips need to navigate through?
Yeah, so there's a lot to discuss about this. And actually, all of the ingredients there are things that I'm not a specialist in, and they must play a very important role in regulating what will turn out to be the niche environment. But there are endothelial cells, fibroblasts, there are macrophages. It's quite a dynamic medium, actually. Yeah. All right, so the next topic, the next section, is what about the mammary stem cells? What is their identity, multiplicity, and sublineage potential? And so for this, we turned to clonal tracing assay, and we used this multicolour reporter system, which I imagine most people here are familiar with. The idea is that we use a system with a ubiquitous promoter to activate genetic labelling of individual cells with a random colour. And of course, it's a hereditary labelling system, so as those cells divide, their progeny acquire that label. And what's nice about the organisation of this system is if you imagine labelling proliferative cells in the tips here, as they expand, they leave behind these indelible footprints in the trailing ducts that provide a kind of memory of what the fate decisions were made in the tips. And the second thing to notice is during this bifurcation process, you can imagine cells scattered between the two outcomes. And so we can look at the statistics of the coarsening of these colours, and we can use this to kind of challenge our ideas about what's going on at the cellular level. So you're not going to be able to see this very clearly, but here again is a mammary fat pad labelled with this confetti reporter system, and it was labelled 3 weeks at the start of puberty. And actually the initial work of this was done by Colinda when she was in Cedric Blancpain's lab. And here is the segmented version of the confetti labelling system, where Colinda's gone through and marked all of those confetti labelled cells at the end of puberty. And so if we zoom in on those regions, if we go down to the rudimentary tree, we have this random labelling of cells that are not dividing. But as we move out towards the periphery, we can start to see these splashes of clusters of labelled cells which are emerging from the proliferation of tip cells left behind in the ducts. And as we move out to the extreme periphery, we see an increasing coarsening, which is compatible with this constant segregation and enrichment of cells. So the first question that we can ask is, are these mammary stem cells sublineage restricted during puberty? And sure enough, when we looked at the clonal level, we can see that clones were either basal-like, entirely basal-like, or entirely luminal-like, which told us that at least at 3 weeks of age, we have sublineage-restricted mammary stem cells that generate the basal lineage and the luminal lineage. We now actually know a couple of things. First of all, we know that those cells are bipotent embryonically, so they become lineage-restricted around birth. And the second thing that we now know is that there's further sublineage restriction. So even in the luminal population, ER positive and ER negative cells are already lineage restricted within that group. So what's the fraction, what fraction of proliferative cells are stem cells in the system? So for that, we can use the following argument that if we label a single cell and ask how many cells does it produce, well, if there are n stem cells, it will contribute on average 1 over n cells to the subsequent subtree. And when we measure this 1 over n and infer the number of stem cells, we find that it's around 90 or around 200 basal and luminal cells, which is very much comparable to the total number of proliferative cells in the terminal end buds. In other words, practically all of the cells in the terminal end buds are actually functioning long-term as self-renewing cells. So they're equipotent in the long term, but what about their short-term potential? And the second thing that we noticed is that if we looked at the correlation of the position of marked cells to marked cells in the ducts, we found that when we had marked cells only at the tips of the end bud, we never got labeling in the proximate duct, or hardly ever. But when they're labeled at the back of the end bud, we always did. And so this suggests that we have long-term equipotency, but the potency changes through a process in which the cells rearrange within the terminal end buds as they grow. And this arrangement is very similar in many ways to what you get in an intestinal crypt, where we showed with Jaakko some years ago that stem cells towards the border of the niche are primed for differentiation and loss, and the ones at the base are primed for renewal and survival in the short term, but long term they function as an equipotent population. And in many ways this system looks like the analogue of a crypt, with a niche-like environment maintaining the renewal potential of these cells in the tip. The difference is, instead of having cells rising out of the crypt and the base remaining stationary, it's these cells that remain fixed and the tips that drive into the fat. So the final question that we could ask from the clonal data is, when we go through a bifurcation, do cells stream into the different sites, or are they randomly mixed through this process? And what we can see is that in both cases, if we look at the distribution of clone sizes as a function of the branch number, in both cases this process would generate an exponential distribution. But the weight of that distribution, the average size of those subclones, in the case where it's streamed, would grow exponentially fast, but in the case where they're mixed, it would grow linearly. And so when we looked at the data, at the subclone distribution, we could see this exponential-like dependence, but actually the growth in the average size is roughly linear over time. And what this is suggesting is that the cells are strongly mixed through this bifurcation process. But what I didn't bring along is an intravital live imaging movie from Jaakko that shows that during that bifurcation process, it's like a kind of washing machine. So if you imagine you have the tip, as the system bifurcates, the cells which are at the tip are folded in to become the border of the new tip. And so the cells are strongly mixed as you move through this bifurcation process. So the second part of this summary is that we think that the terminal end buds constitute a niche-like environment that supports the renewal potential of these cells. It's interesting that within that population, the stem cells are lineage, sublineage restricted, so we think that the basal and luminal cells may also constitute the niche environment that supports each other. It's a point I may come back to if there's time. So during puberty, these stem cells drive this process of ductal elongation and stochastic bifurcation, which becomes terminated when active tips move into proximity of maturing ducts. And during this process, the mammary stem cells switch between the niche border, border region and base, and it reassigns their fate bias as they go. Right. So are these principles of patterning, are they conserved in the development of other branched organs? So we couldn't resist applying this to other contexts, and this is probably now becoming more controversial, but here we go. So, so one obvious place to apply this to is the morphogenesis of mouse kidney. So as we heard not so long ago, kidney develops from an outgrowth of the primary nephric duct, and again it invades through it develops through the process of sequential dichotomous branching, and then a later phase. In a complex, highly complex encoded process, the mesenchyme differentiates into the nephron-like segments that join these filtration units to the terminated ducts. So here you can see segmentation of an E15.5 kidney. So we asked, could this simple paradigm explain the development of mouse kidney? Is there a statistical basis to branching in the kidney? So here we were very lucky. So our collaborator Rosemary Stamponia at Columbia had, prior to our work, segmented the ductal networks throughout embryonic kidney development. And so what we could do is we could just run out our branching, annihilating random walk in 3 dimensions. So now we need one more parameter, because obviously in 3 dimensions you just miss the ducts. Right? So we have a radius at which the ducts will terminate if they meet, the active tips will terminate if they meet a maturing duct. So we have one more parameter to fit, one parameter to fit. So here is a typical simulation of that branching, annihilating random walk. We included some anisotropy to match what you see in the tissue. And we asked, can we predict the segmented ductal networks? So here is experimental data from embryonic day 17, and here is a typical simulation. And we actually can predict rather well the evolution of this branch probability. Now, it could be that there's a stereotypic phase of branching, and I'm very much inclined to trust in the beautiful work of Dagmar. But it's striking that actually you generate this sort of dichotomous branching early on, even from this model, simply because of phase-space volume. You don't collide with anything for a while when you start the branching process. So we can ask, can we predict the distribution of subtree sizes? So here is the number of branches as a function of generation number across the embryonic time course. And remember here we've just got one parameter that we fit, which is just the radius of collision, one parameter. And we can fit, with that one parameter, we can fit rather well the distribution. We can also fit the subtree persistence and the cumulative size distribution of the networks. So there's a huge number of different data sets here that are fixed with a model which has one unchanging parameter. And the other thing that's reassuring is that this is a system that has a phase transition between a phase in which you have 100% tree annihilation and a phase in which you have tree survival. And fortunately, this annihilation radius, the fitting parameter, lines up in this region where we have tree survival. And so the third and final example is the development of mouse pancreas, which again is a fiendishly complex system. So it initiates around E9.5, and there's a huge, a lengthy early phase of plexus remodeling. But the secondary phase of growth is again driven by a program of branching morphogenesis. And so we were interested in this secondary phase of branching morphogenesis growth. And so we used, again, a lineage tracing assay to address whether the statistical basis to branching in this tissue. Well, I'm not going to give an entire talk on pancreas, you'll be relieved to know. But in short, if you look at the distribution of ductal subtrees in development, this is the E18.5 pancreas, when you look at the clonal distributions on this network, they, again, collapse very nicely onto that same branching, annihilating random walk. And there is actually a very interesting difference in the system, and it is the following. So as I say, we did lineage tracing in this system, targeting cells around E12.5 and fixing tissue at postnatal day 14. And what you can see here is that you get entire ductal subtrees which are monoclonally labeled. So we can ask, we have this same kind of branching, annihilating random walk. And we can ask how many stem cells, how many self-renewing cells do you have in the tips? But now instead of having 100, now we have just 2 or 3. So it means that as you bifurcate and elongate sequentially, you very quickly drift to monoclonality, which is why you end up with these fully monoclonal ductal subtrees, or you end up with clones which contain acinar cells belonging to the same subtree, but with no ductal cells. So you've got self-renewing acinar cells that co-travel with those tips, renew with those tips, and when the tips terminate, they generate the acinar. Okay, so are there alternative space-filling strategies? So in this branching and annihilating random walk type model, what we can see is that the ducts pack very inefficiently. But what's an advantage of this system is it's dependent on an unchanging set of local rules. So there's nothing to reprogram during development. It's the same process that occurs sequentially again and again. And so it leads to this kind of perpetual competition between proximate subtrees and this convergence towards this critical life boundary dynamics. But we could obviously do much better if we would have an evolving rule leading to a much more efficient geometric-like patterning. So this would be a much better system to adopt if you really cared about filling space very efficiently. And so we can ask, are there systems that have an evolving set of rules that have a much more efficient strategy? And we heard already about one such example, which is the early phase of lung development, where there are extremely stereotypic rules. In the mouse kidney, other groups have proposed other kinds of stereotyped strategies to develop the network. So there have been proposals that the tips follow a kind of Fibonacci type model, or this half delay model where there's a rule in which you have asymmetrical bifurcations where one tip is delayed. But it's very hard to reconcile these kinds of models with mouse mutants where you see duct-driven termination. And if you run out these rules, you find that they really struggle to capture these very broad distribution of subtree sizes that we see experimentally. So this is experimental data where if we look at in the E16.5 kidney, beyond that stereotypic early phase, we can see some trees have terminated after just a couple of rounds of branching, where others have generated these huge subtrees. In the context of mouse pancreas, other people have proposed that the entire generation of the network comes from plexus remodeling, and a very interesting proposal that late on in development, by driving fluid flow through the ducts, you start reconciling the links. This is a really interesting suggestion, and there's probably a lot of truth in this remodeling process that happens later in development. But this can't be the whole story, because it would be very difficult to understand how a plexus remodeling would generate interpenetrating monoclonal networks of cells. It's, for me, just impossible to see how that can evolve. So the summary is that at the level of the tissue, the statistical organization of multiple models, we've got lots of counterexamples too, but several different branched epithelia are predicted by a very simple self-organizing principle based on a set of entirely local rules which fall under the branching and annihilating random walk paradigm. So in that model, you just have a sequential process of stochastic branching and ductal elongation, but where these active tips become arrested when cells, when they encounter signals from maturing ducts. That's not to say that the cells exit cell cycle. So in the mammary gland, the cells exit cell cycle, but in the pancreas, the ductal cells are still proliferative, and there's local expansion, but they arrest their branching. There's no more branching that takes place, and we think that's because the niche is destroyed at that time. There's no longer renewal of the tips. And what we see, at least in the context of mammary gland and pancreas, that we have cooperative activity of these localized sublineage-restricted self-renewing stem cells, I think they're stem cell-like, that drive this process of ductal bifurcation elongation. We have no understanding of what mediates the branching process. Not that there is no understanding of that, because there are groups, that was a good example, where there's a developing and rather deep understanding of those principles. But in the context of mammary gland and pancreas, we certainly don't know. We don't know. And at the molecular scale, the reason I think this might be interesting is because it raises the question of what are the niche factors that maintain the renewal activity of the tips, and what are the signals that are driving these collective cell cycle exits of mammary gland, some of which we know, and indeed the mechanisms of driving stochastic branching. So, this is fun. So what about other branching structures? There are branches everywhere. And I don't know, so we got tempted by some of these, and then every time we got tempted, we pulled away. So, one of the interesting cases are Pourquie neurons, where if you look at they're very two-dimensional structures, and then you look at the branching patterns here, they look very much like the patterns we generate. I think that this is in a completely different paradigm, because in the case of dendrites, they can grow and retract, and it's the same in root systems, actually. But we did wonder about trees, and actually this is kind of a fun example, because we got very confident. We thought, oh, we'll have a look at trees, and then a colleague in Cambridge brought us down to work very quickly on that. But what we discovered is that the ways that we were thinking are actually extremely popular in the film industry. And there's somebody who Hayat will know very well in Hungary, who actually developed exactly the same ways of thinking in the context of generating these kinds of structures. So these are all artificially generated from a very simple set of non-local rules in this case, because the way the light falls on the lines is very important. But the way of thinking is actually very similar. What I'm not aware of is whether anyone has really segmented trees to really challenge these kinds of ideas. All right, so I don't know how long I've been going, but that's all I wanted to say. But I do want to make some acknowledgements in particular. So I mentioned Jaco van Rienen, and here's Kalinda Schiller, who gathered all of the data on mammary gland. Rosario Samponia actually generated and gave us very generously all of the data on the mouse study. So the pancreas data was actually generated in my lab, so I have a small experimental lab now, and all of that data was acquired by Magda. And the person who I deliberately, until the very end, completely neglected to mention, simply because I wanted to place enormous emphasis on this. So all of this work was actually done in collaboration with a superstar former postdoc of mine, who now has his own group at IST in Vienna, Eduard Haneso. And when I say that he contributed to the work, I do not mean that he did all of the calculations, which he did, but actually he contributed vast amounts to all of the conceptualization of all of this. So he was absolutely instrumental in every facet of this work. And then the last thing I think is a bit of fun, but I can't resist doing that. So of course, the special thanks to Reviewer 2. And that, so actually I feel very bad about this, because actually the truth is I really like Reviewer 2, because Reviewer 2 was great, because it was a reviewer who provided a set, unfortunately a vast set, but a set of absolutely clear scientific questions which you have a chance to answer. So for this, I feel very bad, because I really like Reviewer 2, and I think she is wonderful. Except for this remark, which I think is a very helpful one in this particular community, which was the following. Whilst universal, and probably right, worked against it. And I think that really, I think we completely understand the limitations of what we've done. There are huge degrees of complexity which are operating in all of these different tissues, which are actually crucial to different kinds of questions and different elements of the phenomenology. And the lesson that follows here is that if you will just focus on something very coarse-grained, like the large-scale organization, something as simple as density-dependent feedback is sufficient, I think, to describe what's going on. And that doesn't mean that the regulatory programs that affect all of that are the same, or are even the same at some very basic level. So we completely understand that, and I guess this is probably what would be to. Thank you, anyway.
At least in some of the patterns, there seems to be a variable which is the thinning of branches. Does that play any role? In other words, the early branches are thicker, trees certainly. Does that play any role in anything experimentally? I didn't see that in all the images experimentally, but certainly in some. I thought in the kidney for sure, but perhaps...
Yeah, so I think there are sort of two comments. So the mammary gland is a bit unusual because there you don't see that.
Exactly.
Early on, there is this in the rudimentary tree. There's clearly huge differences, but after the sort of sixth generation, it's in in pancreas. This is very hard to tell because we know that the cells that are laid down in ducts, those continue expanding locally. So then you can ask, well, what conditions the final sizes of those ducts? And probably, what is his name? The guy in Colorado who proposed that fluid flow, the initiation of fluid flow may play a strong role in conditioning the final sizes of those ducts. And he's probably right about that. So I don't want to make a sort of, I think the question of what is the final ductal size distribution, that's, I think, something which is not conditioned by that early phase of laying down the pattern, but is much more associated with the subsequent remodeling that takes place.
And it's happening much later than the morphogenesis of the branching pattern.
It's happening, we think, much later than the decision of the cells to arrest the bifurcation process, but it's obviously contemporary with the development. So these processes may well be taking place while there's still expansion on the boundary. And again, fluid flow may play a key role in all of these different contexts.
So my question relates to the connection between the molecular scale and the cellular scale. So you very nicely demonstrated this unipotent stem cell and also this movement of cells in the cathedral. I was wondering whether this may help to understand basically how they shut down at once. Actually. So maybe it's not
So
naive, but I was wondering whether this is part of the mechanism that helps them to shut down, or whether it's completely unrelated.
So let me give two comments. I won't answer your question. Let me give two comments which are kind of equally superficial, but So the first thing that we know is that TGF-beta is key to affecting cell cycle exit. So one of the experiments that Jaco did was to put in TGF-beta soaked beads into the mammary fat pad and check that the tips terminate on those TGF-beta soaked beads. The second thing that we did, because we wanted our paper to be published, was, of course, we did single-cell expression profiling, which revealed huge heterogeneity of gene expression of cells in the tip. So if you looked at that, it was just a kind of storm of differentially expressed. You could pick out, something like 8 different clusters of the tip. But what we see functionally is that heterogeneity, a lot of that heterogeneity is a fake. That over time, those cells function in the long term as an equipotent group, even though, as a snapshot, we see this expression heterogeneity. So how all of this integrates together, again, I think that this is really a fabulous question. It's at the heart, of course, of what Dagmar is studying, and it's what we want to get to too. So we think, in the context of mammary gland, that what constitutes the tip is that you have an environment that maintains the renewal potential of cells in the tip, but you have factors from the trailing ducts that are actually driving cell cycle exit of the cells at the back of the tip. So it's like the tail is chasing the tip.
I was wondering about the capillaries, because they
anastomos, so in this case they manage to make such networks and to link to each other.
So you mean by vasculature?
Yeah,
Yeah.
vasculature,
So vasculature. I'm not. There may be people who know a lot more about vasculature than me, certainly in the room. Vasculature is much more like a plexus remodeling type of process. So there you do get lots of loops early on in development. You've got huge amounts of cell migration backwards and forwards between the pioneering tips, and probably there the flow is very much conditioning the final network organization. So I'm confident that this particular paradigm won't work, but the way of thinking might be quite useful.
Just on the kidney as a comment, I mean, obviously it's controversial, but I think, it's gonna be really stereotypic. I mean, at least in the early phases, so random or not, but I completely accept that you say, well, it's not about angles, whatever, and random or not. It's all about density. And I think there it's kind of a moot point maybe, because I think the way people look at it is that the limiting factor is the mesenchyme. And so if you have too little mesenchyme, you're gonna stop. And as I said, you can also replace the kidney mesenchyme with the lung mesenchyme. You're going to get a very different branching pattern where the kidney branches are not moving into each other. But if you have a limit, in terms of the amount of mesenchyme you have there, you stop. And it doesn't depend on the branches meeting. So in the natural case, you're going to meet because of the angles, and so that is the condition that you're imposing, but at the same time, you're filling the space. And so that's the condition that maybe the molecular biologists are putting, let's say the mesenchyme is determining the determination.
Yeah. Okay, so I mean, there's a couple of comments about that. So we're saying that there's density-dependent feedback that's mediating the termination of the ducts. But which are all of the components that are involved in density-mediated termination? There can be many different factors, including, of course, mesenchyme playing a role, nephrogenesis playing a role, and so on. So we're looking. It's a bad thing. We're looking at a very coarse-grained level. We have looked at mutants. So if we look at vitamin A deficient mice, we've also Rosemary has also segmented the ductal trees in that context. And we can see that that the organization is very different from the wild-type mice, but entirely captured just by changing that relative rate of bifurcation to elongation. So again, it predicts extremely well the perturbed context. And there are other mouse mutants where the kidneys terminate very early. And those are very much consistent with crossing that boundary where the ducts terminate on themselves because the branch rate is too low. Very much consistent with just crossing this phase diagram. So, at the end of the day, it is possible that there is a completely different mechanism that generates this incredibly complex statistical organization. But my guess is that if there's a completely different mechanism at the coarse grain level, it will boil down to this density mediation.
So that's what I'm saying. I think the molecular one is going to be by the mesenchyme, but it basically corresponds to the same thing of density effects.
Then I think we're just agreeing.
Yeah.
Yeah. Then I think we're just agreeing.
So I'm having a little bit of difficulty understanding why dimension isn't important. And I think it must have to do with this radius of interaction going to zero being a singular limit, perhaps. So if I go to zero, then I won't in 3D, if that radius goes to zero, then in 3D I won't ever intersect. And does that somehow matter? Because the reason
that is, of course, a rather singular point, of course. Yeah, I mean.
Well, I wasn't. No, it's not immediately clear to me.
R equals 0 is a special case. Yes.
It wasn't clear to me that if you, and the reason I bring that up is because if I have a thinning of my branches, then it's not clear to me that you wouldn't maybe go towards that limit if you get thinner and thinner. So that's, I think, the question that I'm asking.
Well, okay. I mean, I think there are sort of two aspects of this. So we think that what's controlling that radius is the secretion of different, morphogenes, where what's controlling that radius is essentially the diffusion length of TGF-beta in the case of mammary gland. So I think this question of ducts thinning down to nothing, which is a very theoretical question because that's of course not what we see. But this question of thinning down to zero doesn't mean that that radius disappears. In the context of the fact that the dimensionality is not important, I mean it's not important at the level of the mean field. And that's very easy to see because if you imagine you just form this front of active tips, you know, every time they bifurcate, of course you will collide with each other. Whatever is that, you know, however small that radius is, when it's non-zero, eventually you drive through, eventually you hit something. So you always end up localizing to a front, and at the mean field level, the dimensionality just doesn't matter. If you're close to the critical point, you know, then of course.
So you mentioned at the beginning of the presentation that side branching is initiated during estrous cycles and presumably during pregnancy. I was wondering, have you looked
That's a
whether the
good
same
question.
rules apply there? And
Yeah.
also, how do you form a new tip where something's dead? So is it the same process in reverse?
I mean, the first question, have we looked at alveoli genesis? And the answer is unhelpful. So reviewer 2 asked us to make sure that the cells that generated the clones were also the ones that were generated in the alveoli, and indeed we checked that, so they do. But the question of which cells do that and are competent to do that, we know from long-term BRDU incorporation that all cells have the capacity to contribute over the lifetime to the generation of new alveoli. But there's very little understanding of who decides to do that at any given time and how many cells are involved in the creation of those alveoli structures. That's not understood. We haven't looked at that, and we should. But I didn't do those experiments, so unless Jaco does, we won't. And the second question was about tip, ab initio tip.
So in your model, a growing tip deactivates when it comes into proximity with an existing duct.
Yeah.
And so I'm trying to think about how do you initiate a new tip from a region of the tissue that's sort of deactivated, assuming that what deactivated it.
No, actually, so there's a really So the question of how those emerge from the rudimentary tree-like structure, I think, is a really interesting question. It's even more interesting in the context of the pancreas, where you have this enormous plexus, out of which have to emerge these minority populations of tip cells that then propagate. We don't know. This is we would really like to understand that transition from this kind of plexus-like organization or the very rudimentary tree-like structure into that phase of branching morphogenesis. But once those tips get localized, then it's deterministic in that setting. There was no side branching. Once those cells have left that compartment, they're not competent under normal conditions to reinitiate the tip branching process.
It's a very naive question, but I was wondering whether the type of network that you generate with those things have some particularity in terms of transport of liquid inside, or whether they are just here to, I mean, in terms of function, whether there is
Yeah, so this goes back to this question of whether you have significant remodeling when you start driving fluid flow. And I think in the case that maybe the mammary gland is very special because you tolerate all of these large vacancies because you can fill them in with alveoli when you really need them. So there you really do get the space filling taking place when, in pregnancy, you fill all of those gaps. So I think that the fluid flow has less role to play in remodeling of the mammary gland in mouse. That's what it looks like. In pancreas, it may well be that fluid flow is crucial. And I think this Copenhagen group has actually been very careful with this, and I trust them. I think that they're probably their right.
So there's obviously a variation of the branch structure, like even in human population of the mammary gland. Also, you mentioned the trees with the branch structures. And if you actually go upside down and think the rule applies, what is the most practical, easiest way to extract like information to compare whether they are different in terms of the simple parameter that you are making?
Sorry, the difference between what and what?
So, for example, like variation of mammary gland, like branching across like samples,
Yeah.
instead of really segmenting everything.
Oh,
Is there like, if you believe that it's following the same rule, like that you for example, a tree, like you said, like you can't segment it, but is there an easier way to actually see between the trees?
Oh, you mean a tree?
Yeah.
Is there an easy way to do that? I don't know. It's a very good question because we're wondering about that. Because we're, of course, really curious about human tissues, which, of course, are hard because of their size and everything else, accessibility. They're much harder to segment. So what we're wondering about is about the analog of clonal tracing. So if we look at the acquisition of mitochondrial DNA mutations or acquisition of somatic mutations, can we use the statistics of that to reverse engineer what were the rules? So we're playing around a bit with that at the moment. And then there's a group in France that we're working with who's actually segmenting ductal tissues in fetuses. It's very kindly producing. I mean, it's a byproduct of his core activity. Alain Chateaudal is producing data which we can also start looking
Okay, so let's thank Dan. So for being able to comment, I just need to sort of head count of the extra.
I'll draw that.